Hello students, welcome to today's Life Sciences lesson for grade 12. And today we are looking at a process called DNA replication. So what is the meaning of the word replication? Replication means making an identical copy of something. So DNA replication is the process by which an identical copy of DNA is made. Do you recall when DNA replication occurs? When would a copy of DNA need to be made? This happens before a cell divides. So if you recall from grade 10, when we looked at the cell cycle, it would be before your mitosis or your meiosis is going to take place. And that part of your cell cycle is called the interphase. So during the interphase, your DNA makes a copy of itself before you reach the phases of cell division. And where does this DNA replication occur? DNA is found in the nucleus of a cell, and that is where your DNA replication is going to occur, right? We think of your chromatin network, so that's your DNA, and you're going to make a copy. Now we want to know, how does DNA replication take place? So we are looking at the actual process, right? So firstly, we need to remember that enzymes are involved in this process. Secondly, let's look at our diagram. What do we see happening to this DNA molecule? We know that DNA is a double helix, but what we can see here, and this is the first step of the process, your double helix unwinds. It takes on that ladder-like appearance. And then your weak hydrogen bonds that hold the two strands of DNA together are going to break. And when they break, your two strands of DNA separate. And that's what we can see on this diagram. Sometimes we use the word unzip when we talk about the hydrogen bonds breaking, and then your two strands actually separating from each other. So let's look at our next diagram, right? We can see our double-stranded DNA in green, and then we can see the next step after the hydrogen bonds have broken, and you've got two separate strands of your DNA. Each of those strands are going to act as a template to make a complementary strand. So think of a template as something that another molecule is going to be built on, or another portion is going to be built on. So you can see your two original strands in green, and notice those little orange structures, and you will recognize the names of the nucleotides on them. So you have free nucleotides that are in your nucleoplasm. And these free nucleotides will become attached to complementary nucleotides on the DNA template. And this process continues until you've built an entirely new strand, and that becomes bonded to the original strand. So you can see that quite clearly in the last step. You notice there are two templates in green, and then notice that there are two strands in orange. So the green are the old original strands, and the one in orange are the new strands that are formed. And now they will be bonded together by your weak hydrogen bonds. And then if we look at our next diagram, you can see that double-stranded ladder-like shape changes, where the entire molecule becomes coiled. So you notice you have your double helix DNA. So what's the significance of DNA replication? Why does it take place? If you remember when it takes place, that will give you the answer. We said it occurs before a cell divides. So the purpose of the DNA replication is to ensure that when cells divide, the daughter cells will have their own copy of DNA. If DNA replication did not occur, you wouldn't get sufficient DNA to actually form your daughter cells, right? And we find in the process of mitosis, the DNA replication ensures that your daughter cells are genetically identical to the parent cell, right? And here we have some diagrams representing that as well. Right. Well, students, that was the process of DNA replication. Now, let's just see if you've understood the process. So I'm going to ask you a few questions, and you must see if you can answer them, right? So I'm first going to give you a simple question, right? DNA replication results in, this is a multiple choice question. A, 2 completely new DNA molecules. B, 2 DNA molecules that each contain a strand of the original molecule. C, 1 new DNA molecule and 1 old DNA molecule is conserved. And D, 1 new molecule of RNA. Right. So which is it, student? What does DNA replication result in? It's option B, 2 DNA molecules that each contain a strand of the original. Remember that you had your original DNA and they separated, and each one was a template, and you built your new strand. So each new strand that forms, each new molecule that forms is going to contain a strand of the original. So it's your option B. Let's look at another question. Right, and we're going to look at some diagrams. And this question says, below are 3 steps in DNA replication. Right, so let's look at them, right? They're labeled step 1, step 2, and step 3. And remember, in a test or an exam, you can be tested on one of the diagrams, and you need to know what's happening, what's going on, what would have happened before, and what is going to happen after, if that applies. So let's look at our question. It says, explain what is happening in step 1, step 2, and step 3. And each one is just for 2 marks, right? So let's explain what's happening in step 1. So look at step 1. What do we see? You see your double-stranded DNA, and you can see the 2 strands are beginning to separate from each other. And since it says explain, you need to say, the 2 strands of DNA separate from each other because the weak hydrogen bonds holding the nitrogenous bases together break. Right. So that's your answer for step 1. Let's look at what's happening in step 2 and look carefully. Right. You can see some distinct DNA strands. Right. You can see portions that are double-stranded. And what you can also see floating around are nucleotides. Right, so let's explain what's happening in step 2. In this step, free DNA nucleotides are becoming attached to complementary bases on each of the DNA templates. I'll say that again and watch the diagram, right? Free DNA nucleotides become attached to complementary nucleotides on both your DNA templates. We can see that clearly. Right, and let's look at step 3. In step 3, you will say 2 DNA molecules have formed, and each of those molecules have 1 strand from the original DNA and 1 new strand. And you could also state these 2 molecules of DNA are identical to each other. If you look at them closely, you can see that. Right. Here's another very simple, basic question. Where does DNA replication happen? It happens in the nucleus, because remember, your DNA your chromatin, your chromosomes are there in the nucleus. So the answer is nucleus. Next question. When does DNA replication happen? Right. DNA replication happens in the interphase of the cell cycle. Remember, this is before your cell divides, right? So it would be the interphase of the cell cycle. Right. One more question for you. Below is a portion of a DNA strand. It says make the complementary DNA strand for the portion that is shown. So look at your original strand, right? A portion, a small portion of it. So that strand has A, T, G, C, A, T, right? So that's what you can see. So we now want a complementary strand, right? So let's go, right? So you say it along with me, right? So when we start, we're going to have as complementary T, A, C, G, T, 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 and A, right? So if we can read that again, it will be T, A, C, G, T, 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 A. Right, students. I hope you enjoyed the lesson on DNA replication. Please practice the process, say it out to yourself, write it down, draw the diagrams, and I will see you the next time. Take care.